Antibody therapeutics are often initially developed in non-human systems like mice. But these proteins can look foreign to a human immune system. This can trigger an undesired immune response against the antibody itself. The purpose of humanization is to change the antibody molecule such that it looks more like something the human body would produce, therefore decreasing the immunogenicity risk. In CDR grafting, the standard humanization approach, you transplant CDRs onto a homologous human framework. You usually lose binding affinity in the process and spend time adding back mutations to recover it, without a principled way to know which positions were safe to change in the first place. I'm Janneke, machine learning scientist at ENPICOM, and here's how we think about solving that. With every mutation you introduce into the antibody, you risk affecting the binding affinity or other characteristics of the molecule. Therefore, you would ideally mutate only those residues that have the biggest impact on the immunogenicity of the antibody. So how do we know which residues have the largest impact? In fact, how do we know how immunogenic a molecule is at all? For this, we can use a proxy based on protein language models. By training a protein language model on a large set of human antibodies, it learns what these antibodies look like and can tell how out of distribution any given antibody is. This metric indicates the humanness of the antibody. As can be seen here, the amount of anti-drug antibodies that a therapeutic antibody elicits correlate with the humanness of the therapeutic antibody. The fewer positions you touch during humanization, the less you risk affecting binding and other developabilities. So the question is which positions you actually need to change, and which ones you can leave alone during humanization. Positions crucial to binding stay locked, whether that's CDRs, Fournier regions, or other positions identified as critical depending on the project. We then use the protein language model to determine which positions have the biggest impact on humanization. These are prioritized for substitution. Each substitution is conditioned on the surrounding context of the antibody molecule. What counts as a good change depends on the neighboring residues. The result is a humanized sequence whose binding is still intact with a small added distance from the starting sequence.